The parable of the elephant and the blindfolded scientists is a reminder that no single sensory modality or frame of reference is sufficient to fully capture reality. And the opening sound is a reminder of the remarkable capacity of the human auditory system to extract information from a complex soundscape. And these are some of the reasons that we've been using auditory analytics to help us study hydrogen bond dynamics in proteins and synthetic polymers. Auditory analytics is essentially the scientific method, augmented by data sonification and analytical listening. In this context, sonification is not an isolated goal in itself. It's an integral part of a larger research cycle that includes collecting raw data, deriving data sets, designing multiple mappings of data to sound and to visual representations, analytical listening, hypothesis formation, and building tools. We always end up going around this loop many, many times, generating many different mappings, building new sonification tools, and occasionally collecting additional raw data before going around the loop again. Okay, we are on slide 6, 7, 6.
So I love working with Carla and Kurt because unlike Bruce and Scott and Carla and others here, we don't investigate sonification per se. I don't build sonification tools, but they do. And they build very nice sonification and visualization tools that have proven to be useful in the research that we do. So what are we researching? Next slide. We look at proteins. In particular, we're interested in how proteins gain their shape and their function. So a protein like, for instance, this thing that we call WW over there, starting out on the right-hand side, is in some local minimum of the energy, oh, don't advance it yet, is in some local minimum of the energy landscape, and eventually ends up on the folded side, on the top left-hand side, or in this case, it makes this triple-stranded beta sheet that sort of is like these, 3 strands that are symbolized by these arrows. Now, it turns out this kind of folding is really important. In the case of WW, literally for survival from minute to minute. So in every one of your cells in your body, this protein interrogates another protein about 1,000 times per second. And as long as that interrogation comes up with a yes in the signaling, you live. And if not, your cells undergo a process called apoptosis that instantly kills them. Now, you might think, oh, why do we have something like that? Like, who wants to have like a clock that ticks 1,000 times per second and you die if it stops ticking? Well, actually, it has good uses. So for example, for those of you who had to walk over here, even on a cloudy day in New York with the sun not out bright, there's scattered UV light that hits your skin, and you will have on the order of 10 to 20 lesions by the time you have walked 10 or 20 minutes coming over here. It turns out that the cells on the surface of your skin recognize something is wrong with them and turn on this mechanism and kill themselves, and before they develop into cancer cells. Sometimes you get unlucky, and one of the cells that gets hit, it's the actual mechanisms that help the cell recognize that it should be dead that get hit. And then, of course, the cell is immortal and can turn into a cancer. Right? So death can be bad, but death can also be good if it's the bad guys that get killed. So what do we actually study specifically? So we're interested in how this unfolded protein, transfers over from the unfolded side to the folded side over a barrier in energy to a lower energy state. And there's 2 things I want you to think about as we talk about it. The amino acid residues that this chain is made out of can be classified into 2 types, and one is called hydrophobic or oily. And those oily amino acids actually want to be separated from water and just glob up among themselves at the interior of the protein in the core. And this is one organizing principle. So you want those things to end up in the middle, just like an oil droplet will separate from water. And the other thing they can do is they can make hydrogen bonds. And these hydrogen bonds organize structures such as these beta sheets that you see there. So basically, an oxygen atom and a nitrogen atom might end up having a hydrogen between them. And what we're really interested in our particular research is not even, what do the folded proteins look like? There's programs like AlphaFold that can take care of stuff like that. We're actually interested in how does this hydrogen bonding change and contribute to forming that structure as the protein is making that transition over this energy barrier to find this lower energy state that's the folded state. So a graduate student of mine and me, when we started looking at this research, we started with visualization. And what we ended up doing is we ran these simulations, or actually we used simulations that we ran together with D.E. Shaw, a place in New York that does these kinds of simulations on supercomputers. And these simulations contain tens of thousands of atoms, all the water molecules and the protein molecule moving around, and we solve Newton's equations, kind of the way you would if you did a galaxy simulation and you just solve Newton's equations and see how the thing evolves in time. And then what we try to do is we are interested in the hydrogen bonding patterns with the protein. So we got rid of all the ones that are not near the protein, like we try and simplify the visuals, and then started looking at those simulations. So here's one of them. And the protein will fold at the end, and it'll be shown by those little arrows, but they're not really there in reality. It's just something to indicate those beta sheets forming. And we were trying to make sense of what's happening with those little red balls that are oxygens, and the little white ones are hydrogens forming water molecules. And I'm sorry to say, but we looked at this stuff for a long time, and I don't know if you can see a deep pattern in there, but we weren't able to tell what's going on. And the reason is visually, you have really hundreds of those hydrogen bonds that form and break in picoseconds and are remade all the time. And your eyes, there's correlations between them that are non-local, but your eyes are not really very good at looking at the right spots where something might or might not be happening. So this turned out, we tried that for a while. That was really not a very good way that we were making any progress.
But it turns out that's exactly the situation where the auditory system can really shine, because a few minutes ago, you guys were able to extract information from multiple sound sources. There were elephants, birds, distant echoes, the sound of a breeze, and you extracted that all in a matter of seconds. So the auditory system is really good at tracking multiple sound sources, especially when they are separated in time and spatial location, and they occupy unique frequency band. So we tried to map each hydrogen bond event to a sonic event, a grain of sound where each grain is localized in time, in pitch space, and in stereo position. So now we get to the analytical listening stage of the cycle. The thing about analytical listening is, one, you already know how to do it, and two, everybody gets better at it with practice. So for example, listen to the direct H bonds forming between different parts of the hydrogen of the protein with itself, and raise your hand when you hear a dramatic change in the soundscape. So you've just heard with your ears the distinct signature, hydrogen bond signature of the protein folding. By listening to subsets of amino acids and H bond types, we immediately heard an association between H bond dynamics and how quickly a transition occurred. Then we spent a lot more time confirming what we'd heard by automating the identification algorithm and performing standard statistical analyses. Our ears had already told us there was something to find. We just confirmed it with modeling and statistics. Ironically, in the process of kind of self-reflecting on how we identified those H bond patterns by ear, we came up with a new visualization. So we ended up with a visualization of a sonification. And to their credit, this is now slide 11. To their credit, the editors at PNAS gave us permission to link directly to sonifications from within the body of our paper. And we recently got permission from Wiley to do the same. So if every one of us using sonification starts making that request, I think it could help elevate the status of sonifications to the same first-class status enjoyed by tables and graphs in scientific publications. Over the course of this project, we've been building up a software framework of reusable modules for sonification and concurrent visualization. This was to make it easier for us to experiment with multiple alternative sound mappings. For example, this part of the graph defines a sound generating algorithm, and it's modular, so you can swap it out with any other compatible algorithm. To specify that a sound parameter can be controlled by data, you set it to a variable. So in this case, the frequency of the oscillator is set to the variable log freq in units of pitch. The data mapping environment is part of that same signal flow graph. To define a mapping, you declare the target variable, log freq, and associate it with a block of code to execute on each row. This example reads a value from the endpoint column, and it has several options for normalizing, warps the value through an adjustable curve for log, linear, or exponential perceptual scaling, scales the value by pitch deviation, and offsets it by a pitch base. So this allows researchers to adjust the range of the values while listening, either to bring out aspects of the data or to place the sound in a range where their own hearing is most acute. So this is slide 14, and Martin can introduce these guys.
So you've heard a little bit about the internals that go on in the software as well. So let me talk about a different project that we're in the middle of working on with Martina Havenet and her postdoc Wanlin Chen. And this one we also sent to PNAS, and it's coming back with favorable reviews. You might not be surprised to hear, though, that the reviewers have no comments about the graphs in the paper. But 2 of the 3 are asking, like, how do you know that sonification is real? As usual, right? We've heard about this a number of times, but we think we have good answers to those questions. So what's this project about? Well, in some ways it's a little similar, in some ways different. So there's a polymer called PNIPAM. It differs from a protein in the sense that it has 30 subunits, but they're all identical. So in a protein, you have different amino acids. Here you have just 30 of the same things. And it also behaves a little differently when it sort of becomes compact, or if you want to call it folds. It turns out proteins behave more intuitively. They're folded at low temperature, and if I heat them up, the thermal motion causes them to fall apart. This polymer is actually unfolded at low temperature. When you heat it up, it contracts and forms a compact globule. Okay, so a little counterintuitive. So one of the things we're interested in, how does hydrogen bonding contribute to this counterintuitive behavior? And again, you see the tool set that we had built up. And the first thing we did, since we already were wise that looking at water molecules to look at hydrogen bonding is not going to tell us the answers, we went straight to a sonification. And we had a hypothesis, which was that just like in proteins, direct hydrogen bonds between these side chains of this polymer now are actually what's holding the polymer together and organizes it into its shape. So here's a sonification of that. And you can tell there's something going on once in a while. That's it, not much. So actually it turns out our idea here was wrong. It's not the amount that you get of direct hydrogen bonding in this polymer is just not sufficient to explain the stable structures and compact structures that form. So there had to be something else going on. So we threw out our initial guess and tried to come up with other guesses. Now, to do that, we actually went over, next slide, to visualization. Nothing wrong with that. Go on to the next. There we go. So the kinds of bonds we had talked about initially, or guessed might be the case, are these direct bonds. You have a nitrogen in blue, you have an oxygen in red there, and that little dashed line goes to a hydrogen that connects the two. So that's a direct, so-called direct hydrogen bond. So what we did is we looked for anecdotal evidence. There might be other things that people hadn't talked about in the literature. And we actually found something that was never mentioned anywhere in the literature at all, which we call SCB here, or side chain bond, where actually two nitrogens, again in blue, and hydrogens line up parallel to one another instead of like this, which is a normal hydrogen bonding pattern. And then the other thing that we thought might be a candidate, and we sort of looking around the images, you could find them, are things like water-bridged hydrogen bonds. So there you can see there's an oxygen red at the bottom and a nitrogen in blue at the top, but they're not bonding directly to one another. There's a water molecule stuck in between, an HOH, that white, red, white molecule. The problem, though, again, with the visualization is we can tell that these things are there, but we can't tell from visualization how they're correlated. Again, there's hundreds of them showing up in various places. So we have some anecdotal evidence that this might play a role, but it wasn't obvious which ones are important or not. So we constructed then, again, a sonification, and you can run it if you want, based on a reduced dimensional space where we took a large number of variables that have to do with the solvation of the system, the compactness, and we'll do that soon. And what you're seeing there is actually a trajectory of the system in this reduced dimensional space and a sonification, which you'll hear again on the next slide. The yellow side is the compact side, the purple side is the extended side, and those turquoise or greenish transitions in between, it just went through one. That's actually when it transits over between the compact state and the extended state. Let's look at the next slide. So Carla and Kurt constructed this very nice user interface that allows you to look at the dynamics in this reduced dimensional space. She mentioned the piano roll that we had come up with, which is a visual representation that actually has the residues on the vertical axis. So from residue number 1 to 30, in this 30-mer polymer, and time on the other axis. It's color-coded so that hydrogen bonds that form water-mediated different distances are coded in different colors. So you can tell when interactions are local and when they're non-local. And if you want to run this one, listen to the sound, the difference in the sound on the purple side versus the yellow side. Compact. Here we're completely unfolded. So what took some training is you could hear the difference between the yellow and the purple. But actually, if you listen to these things, and I spent weeks, doing this, playing around with it, you actually really get used to the scale mapping, the panning mapping, the various sound mappings that were used, that you can hear fine details about, there's like a correlation where this direct hydrogen bond can only form if this water-mediated one forms first, things of that sort. So what were the conclusions we came up to? Direct bonds are not the primary stabilizing force in this polymer. Instead, it's actually water bridges that turn out to be the important thing. And the other thing we found along the way is there's a number of traps or intermediate states, and that sometimes bad bonding patterns can actually hinder the direct transition from the extended state to the compact state.
So we discovered that discovery actually requires active exploration. You need to be able to build new tools on the fly as questions arise. And that's where dynamic programming languages like Smalltalk and Python can really shine. Also, ideally, sonifications and visualizations should be interactive so researchers can explore the data on their own terms. In the end, no single data set, visualization, or sonification can ever provide a complete map of a phenomenon. Instead, scientific facts are established progressively through a cascade of evidence: texts, equations, statistics, sonification, visualization, all working together to transform data into a cohesive and cumulative argument. And data sonification and analytical listening form an indispensable part of that cascade of evidence. Thanks.
Thank you very much. That was really interesting. Does anyone have any questions in the room or on Zoom? Scott. You can summarize the question here. Have we got a sorry? The handheld one's working.
This is an inherently complicated 3D problem, as you know. Has anyone explored combining the sonification with VR or AR technologies so you can see it in actual 3D with or without the sonifications? Because your animations on 2D did look completely chaotic, but it might help if it's projected and you can actually explore 3D space. Back in the day, we had cave systems that were supposed to be replicating that.
So we did that. And BMD, which is the software where you do this visualization, allows you to rotate the molecule while you look at it. We didn't do stereo glasses, but the rotation gives you a pretty good idea of how it looks from different. The problem is just there's multiple hydrogen bonds that are visually separated on the sequence. And when we look with vision, I can focus on you, right? And I can tell that you're there. But if I really want in high resolution to know where you're wearing it, I need to actually look at you. That's really the problem. So you have these delocalized events. Folding is an emergent phenomenon that in the end occurs because hydrogen bonds are correlated in different places, sometimes well separated, but nonetheless coincide, right? Because these two things cooperatively have to happen at the same time for the thing to be able to collapse. So we just didn't have any luck with that. Whereas listening to the sound was a much easier and better way of doing. In hindsight, we then came up with visualizations that work pretty well in reduced dimensional spaces or things like that. But we already knew what we were looking for at that point.
Great.
Adam?
To your last point, Martin, it seems like you knew what to hear for as well, right? Because you built this sonification software with some a priori definitions in mind about what you were looking at, right? And so I'm just trying to I think this is amazing, right? But I'm just trying to get.
Yeah. That's right. The good news, though, is, so let me take the WWM as an example. What we ended up doing is just doing granular sonification of many of the residues or amino acids in the protein and have, as Carla was saying, is very important, a user interface where you can turn on only the direct ones, only the ones between N and O, only the ones between N and N. And we actually did, so there was preconception in the sense we did believe there were hydrogen bonds. But what really worked for us in the sonification process that was difficult to do by visualization is I can just turn the N-ends off and listen only to the other ones. And I really did spend, and the students who worked on this, we spent hours listening. It took us a while to really learn, what to listen for. But we also spent hours on the visualizations to try and see, and it really just wasn't working. So of course, there's always a model that you build, a priori. And in this case, it was having a granular sound that corresponds to various kinds of hydrogen bonds forming. But the neat thing was that turning them on or off, my ears can actually tell when I turn one off, it's gone. When I turn it on, it occurs always after another one. It goes bum, bum, never bum, bum, right? And so you learn those things. And that really is what got us to find patterns that we didn't actually anticipate in the data.
And I should clarify that the difference between the panel that we showed and the modular framework. I mean, the modular framework is very general, and then we made specific, multiple ones. we tried many different panels until we ended up with the right panel for doing the test.
Amy, go ahead online if you've got
Yeah,
a question.
thank you. Thank you very much. Thank you for sharing your work. It's very interesting, fascinating, really. I was wondering if we could back up, zoom out a little bit, and if you could, if you're willing, share a little bit about how your partnership developed and how the work was supported financially. For example, did you envision this level of partnership from the beginning and write a grant proposal and get funding to work together, or was it no? Okay.
This, as many wonderful collaborations do, kind of emerged out of by chance, or because of the pandemic. During the pandemic, a lot of us couldn't. Well, no one could travel during the lockdowns. So there were a lot of people who had a little bit more free time, I guess. And we, one of our, a colleague, a mutual colleague, introduced us to Martin, and how did we initially start out? We started out looking at something totally different, and it evolved. And no, we don't have any funding. In answer to your question,
So,
I mean, other than
yeah, I mean, to be fair though, Kima is a company, and so it's their, they fund themselves, right? That way. And the way I did it on my side, I have a grant to investigate protein folding. In that grant, it doesn't say that I must make graphs or that I must listen to sounds. It says the problems that we're supposed to, solve. And that's why I was saying yesterday, just do it, right? Because in the end, people who look at research grants don't care whether you make plots or sounds or whatever. They might be a little more sensitive about sounds because that's not such an established thing yet. But if you tell them, I want to understand hydrogen bonding and protein folding, you don't say anything about it. That's perfectly fine. In hindsight, this is what ended up working well for us, and the reviewers loved it when we wrote it up. I mean, it depends on, I guess, the reviews you get, but sometimes you get lucky and you get people that go like, hey, this is like weird and different. Yeah, I like this. So.
And the PNIPAM project was kind of spontaneous.
Yeah.
You were visiting.
Yeah, I was visiting Germany, collaborator, found out that they were doing these simulations. They didn't know what to do with them. And so we said, yeah, I think you could do this. And I put them together with Carla and Kurt as well. But, and so in the end, like I think all research, it kind of grew organically. And we didn't even necessarily advertise, we're going to use sound. But we did it, and places like the NSF and NIH and the end, they care about the result. NASA probably as well, right? I mean, there's a scientific problem, they want to see it solved, or at least they want somebody to try and see a solution. And after that, you kind of can do, you do it the way that you think works best. So, I mean, after all, hopefully our funding people think that we're adults enough, right, that we can make the decisions about how exactly we're going to plan the research work so that we can get the most interesting results out of it.